morning.
Good morning.
I hope you've seen that I have posted a study guide on Blackboard, so I encourage you to take a look at these questions that are referred to in this study guide. Okay. So remember, we'll have the first exam next Tuesday. I will be holding my last office hours today before the exam. So if you want to talk with me, you may want to take advantage of that. That's the last opportunity to talk with me before the exam. I will not be holding office hours on Tuesday. So if you have to talk to me, come today, please. The format of the exam will be Scantron-based multiple choice type questions. So as I indicated before, I guess you don't have to draw any structures, but I encourage you to be able to do that. You may want to pay attention to what I say now, or not. So go over the lecture materials. These are the basis for the exam. Don't just depend and rely on the study guide. Okay, this would be a huge mistake. So what I tell you in class is relevant, and then once you feel prepared for the exam, I would suggest you take a look at these questions. Be aware of the fact that there may be some questions where you have to calculate stuff related to bioenergetics. So there are a couple of questions in the study companion to the textbook, possibly in the textbook itself, can't remember, where you can practice questions like that. So take advantage of that. Do you have any questions about the exam?
Do we need to bring a calculator?
Yes, a standard calculator. Nothing fancy. Okay, so we will check whether or not you have programmed anything related to biochemistry into your calculator. If we find anything, it will be an automatic 0 points, and this may have further consequences. So just a regular calculator and a pencil you will need, and a photo ID. Okay, so you won't need anything else. No scratch paper, no nothing. Any other questions? I will let you know later today up to which point the material will be relevant for next week's exam. And anything I discuss from that point on will be relevant for the second exam. Okay? So, we were talking about regulation of glycolysis, and I told you to remember that there is a relationship between the energetics of individual reactions and the likelihood of enzymes catalyzing these reactions to be subject to regulation. And we see this also in glycolysis, where the three enzymes, hexokinase, phosphofructokinase 1, pyruvate kinase, are regulated also to catalyze hexagonic / irreversible reactions under those conditions that we find in cells, please. We talked a little bit about the regulation of these enzymes. I told you that hexokinase, muscle hexokinase, I should say, or extrahepatic kinases are regulated by a competitive inhibition through glucose 6-phosphate, the product of this reaction. So this is not uncommon that the product of a particular enzymatic reaction, as it accumulates, also is able to inhibit this enzyme. So we see this here, the structural analogy of glucose 6-phosphate. Glucose is the basis for this competitive inhibition. So the product can bind to and block the active side of the enzyme. I talked a little bit about the inhibition of pyruvate kinase, and I said you may want to take a lot of this with a grain of salt, but the general principle that we see here in these names actually applies here to the regulation of this enzyme and many other enzymes as well, namely that if we look at enzymes participating in catabolic reactions, molecules that signal an energy sufficient state have an inhibitory effect, and those molecules that signal an energy insufficient state like AMP and such tend to have an activating effect on such enzymes. And then I also mentioned glucokinase, okay, I forgot about this, where the regulation is different compared to the other isozymes of hexokinase. And we have this transport between the cytosol, where glycolysis takes place, and the nucleus, okay? So the last enzyme whose discussion, whose regulation I discussed was phosphofructokinase 1, and I told you that the most important allosteric modulator of this enzyme is fructose 2,6-bisphosphate, which can be produced from fructose 6-phosphate via a phosphorylation reaction catalyzed by phosphofructokinase 2, okay? And I told you that the phosphate can be cleaved off by an opposing phosphatase activity, so the enzyme can be called fructose bisphosphatase 2. And interestingly enough, both these opposing activities, the kinase, so the phosphorylating activity, and the phosphatase activity, which is the dephosphorylating activity, reside on the same enzyme, same protein, which we can refer to as a tandem enzyme because it has these two activities. And then I told you that whether or not we have the kinase activity or the phosphatase activity depends on whether or not this tandem enzyme is phosphorylated at a particular serine residue. And I told you that this phosphorylation depends on the secretion of glucagon, which actually stimulates adenylyl cyclase indirectly via G protein, doesn't really matter, and this leads to an intracellular increase in the concentration of cyclic AMP, which is produced by adenylyl cyclase from ATP, and then cAMP can activate cAMP-dependent protein kinase, also called protein kinase A, which can phosphorylate a bunch of proteins and enzymes. Among others is this tandem enzyme. Okay. So this is of relevance in particular for liver metabolism. Okay, I indicated previously that the liver is the organ that is most important for homeostasis of glucose in our body. So we can have both glycolysis and the opposing pathway gluconeogenesis happen in hepatocytes, in liver cells, but these two opposing pathways are not supposed to happen at the same time. Okay, so we have regulation of both these pathways, and this kind of phosphorylation and activation or deactivation of phosphofructokinase 1 then ultimately via fructose 2,6-bisphosphate is key also in the regulation of gluconeogenesis and glycolysis respectively. We'll see this again when we talk about gluconeogenesis, and then when we talk about, especially when we talk about degradation, regulation of glycogen synthesis and degradation respectively. Okay, so what we've seen here in this table also is that phosphofructokinase 1 is also subject to a bunch of inhibitors, among others citrate, which also signals an energy sufficient state, and also ATP. Okay, I'll talk about citrate, why it is signaling an energy sufficient state later. But it's, yeah, okay, let's talk about this later. But ATP also has an inhibitory effect, and this is quite interesting if you think about it, because ATP is also a substrate for this enzyme, phosphofructokinase 1. Remember what phosphofructokinase 1 does? Okay, phosphorylates fructose 6-phosphate to produce fructose 1,6-bisphosphate. So ATP is the source of the phosphoryl group. So ATP is a substrate, but then we see here that ATP also is an inhibitor, which is quite interesting, I guess. But the basis of this is that phosphofructokinase 1, which actually is a tetramer, shown here only two monomeric parts of the tetrameric oligomeric enzyme, has different binding sites for ATP. Okay, so the active site obviously has to be a binding site because there we have the phosphorylation of fructose 6-phosphate happen. So the active sites are located here. But then we also have allosteric sites where ATP, as it increases in concentration in the cell, can bind to and then inhibit in an allosteric way the enzyme activity. Okay, and this has following effect on the activity. if we look at such a Michaelis-Menten kinetics plot, the black line, actually the black curve, indicates the activity of the enzyme phosphofructokinase 1 as a function of the fructose 6-phosphate concentrations, fructose 6-phosphate being the substrate. So this is a typical kinetics curve, let's say. But as we see here, if the ATP concentrations actually increase, we have a shift of this curve. We also see some cooperativity here, more than before, as indicated by the sigmoidal curve. But we see that the activity actually has gone down because we would need quite higher concentrations of the substrate to reach the same activity. Okay, so we have a shift to the right and a shift to the right. We look at something like the Km values here, okay, 50% inhibition or 50% maximum activity values here. We would see that these have gone up, Km values, and an increased Km value indicates that we have less binding activity, less affinity for the substrate. And overall, if we compare the same concentrations of substrate, at least over a wide range of substrate concentrations, we have less activity. Okay, so shift to the right means we go down in activity. Okay, so this slide just summarizes, let's say, the more important effectors of the activity of phosphofructokinase 1. So citrate, I mentioned, also signals an energy sufficient state as an inhibitory effect. Just seen what effect increasing concentrations of ATP have on the activity. AMP and ADP, which both signal an energy insufficient state, have an activating activity, which makes sense. And so does this most important allosteric effector, fructose 2,6-bisphosphate. Good. So let's now come to the fates of pyruvate. So pyruvate can be produced in glycolysis, as we've seen. For each molecule of glucose, we produce 2 molecules of pyruvate. And then I mentioned a couple of times before that we have to distinguish between anaerobic conditions and aerobic conditions. So I told you that under anaerobic conditions, all the energy yield that we get out from the degradation of 1 mole of glucose is 2 moles of ATP. So that's the net energetic yield. And the energy that is stored in NADH produced in the glyceraldehyde 3-phosphate dehydrogenase reaction is pretty much lost. Okay? Because we do not have aerobic conditions, we do not have cellular respiration, we do not have electron transfer happen, we do not have oxidative phosphorylation happen. This is something that we're going to discuss pretty soon, what that actually is. So, under anaerobic conditions, then, we have to distinguish between different organisms, potentially, that can produce one or the other pathway of the 2 that are shown here. So if we just focus on humans, we can perform metabolism under both anaerobic or aerobic conditions, depending on tissue and supply of oxygen of certain tissues. So if we, for example, look at contracting muscle tissue, so we typically can perform oxidative metabolism, but then there might be situations, for example, where we have a vigorously contracting muscle. If we run for a long time or sprint for a certain time, our muscle tissue may not be sufficiently supplied with oxygen, so that we switch over to anaerobic metabolism. Okay? And then we would actually have this fate here where lactate is being produced in tissues. Now there are a couple of tissues in our body that actually, let's say, only can perform anaerobic metabolism, like erythrocytes, or there are some that just do that, like retina cells, for example. Then there are other tissues, other cells that like to perform oxidative metabolism and have a very, very active consumption or high consumption of oxygen, like the brain and nevertheless also to a certain extent produce lactate under such conditions, maybe for other reasons. So this is coupled with amino acid metabolism. But then there are also other organisms that actually work under anaerobic conditions and produce also lactate. So we're going to take a look at what happens there in lactate production in a moment. Now there's another possibility, another anaerobic fate that pyruvate can have, which is shown here on the left side, which is the conversion of pyruvate into carbon dioxide and ethanol. And as you probably know, there are a couple of organisms that can do that, among others yeast cells. Okay, so they can, under anaerobic conditions, produce ethanol and carbon dioxide, which for thousands of years has been utilized by humans for beer brewing and wine making and bread baking, okay, things like this. Now, what's the reason why we would perform these reactions that lead either to lactate or ethanol? The common reason for doing this is that these reactions regenerate NAD+, which is needed in glycolysis for the glyceraldehyde 3-phosphate dehydrogenase reaction. Remember, there NAD+ is reduced to NADH. Now, if we just have NADH produced, but have no NAD+ anymore, we would not be able to perform this reaction anymore, and we would not be able to perform glycolysis. And with this, such cells that have to perform glycolysis under such conditions wouldn't get any energy anymore and would probably die off sooner or later. Okay? So the main reason for doing these reactions, performing these reactions, is to regenerate NAD+. Okay, and for that reason, NADH that is produced under anaerobic conditions cannot be utilized for energy production because it's just simply regenerated to NAD+ to just keep going with glycolysis. Under aerobic conditions, which we are going to discuss starting today, first we have a conversion of pyruvate to acetyl-CoA. This is catalyzed by big protein complex located in the mitochondrial matrix called the pyruvate dehydrogenase complex. And then the acetyl-CoA produced in this reaction is channeled into the citric acid cycle. Okay, we'll talk about this later today. But first, we want to focus on these two fates under anaerobic conditions, and we'll start with a discussion of conversion of pyruvate to lactate. So this is a simple reaction where the ketone, so the carbonyl functionality at carbon number 2, is being reduced to a hydroxyl group. Okay, remember, ketone to hydroxyl is a reduction. Okay, we always have coupled redox reaction, so something else actually has to be oxidized, and the something else here is NADH, which is being oxidized to NAD+. And here we have the reaction that we actually need to have. Okay, we have to regenerate NAD+, and this NAD+ produced in this reaction here, catalyzed by lactate dehydrogenase, can be utilized then again by glyceraldehyde 3-phosphate dehydrogenase in glycolysis. So this can happen in our body, and the lactate dehydrogenase that we have actually is stereoselective and produces the L enantiomer of this chiral compound. Okay, so just note that here is the chiral center. We have 4 different substituents surrounding this asymmetric carbon here, and here it's written in the Fischer projection. The hydroxyl group is pointing to the left, so this is the L enantiomer. Okay. Now there are organisms that can also, microorganisms that can also produce D enantiomer, bacteria. Okay, and the occurrence of D-lactate actually in humans indicates bacterial infections. So this can be used as a diagnostic tool for diseases like short bowel syndrome and things like this. If you have a bacterial infection and these bacteria produce D-lactate and you can detect this, then okay, use this as a diagnostic tool. So I just mentioned bacteria. Lactobacilli actually also perform this reaction. So bacteria that we find in milk, okay, and the lactate, which could be L-lactate or depending on the bacteria, actually D-lactate, then leads to an overall acidification of milk, precipitation of milk proteins, and this also has been used for thousands of years by humans for the production of cheese and yogurt and things like this. Okay. Now I'm not quite sure, never found out whether also in this country there was this craze about D-lactate in yogurt, but at least in Germany for the longest time, I'm not sure if they still do that, you would see on yogurts specified that they do not contain D-lactate because it was considered to be bad or whatever. I'm not quite sure if this makes any sense. I don't think it will do any harm, but I think they associated the bacterial infections and the accumulation of D-lactate, if you have a bacterial infection, with the presence of D-lactate also in yogurt for whatever reason. I'm not quite sure if you take it up, if that would have any harmful effects or so. Okay, so in humans, we actually have two forms of lactate dehydrogenase, which can be separated, for example, in electrophoretic experiment here, and these two different types have been named after the tissues where they are most, let's say, prevalent in the pure form, namely in muscle tissue, skeletal muscle tissue, and in heart muscle tissue respectively, and for that reason they have been called M and H lactate dehydrogenase respectively. They also come with different names, but this makes it clear where they occur in the pure form. So both of these forms actually form tetramers, as you can see here in the schematic representation. And so in skeletal muscle, and also interesting enough in the liver, we have a tetramer that is only composed of 4 M monomeric subunits, while in the heart and erythrocytes we have a tetramer that is composed of only 4 H forms of the enzyme. But then in other tissues we can actually have hybrids of different composition that also have different electrophoretic mobilities. With regard to the mechanism, they don't differ. They may differ with regard to regulation, affinity, and things like this, but the overall mechanism for both of these forms is the same, and is shown on this slide, which shows us that hydride actually from the ray side, so the upper phase protein amide ring in NADH, is transferred here to our carbonyl carbon pyruvate. Now this is held in place with the help of charge-charge interactions with an arginine in the active site, and then a histidine residue in the active site actually protonates the oxygen, previous carbonyl oxygen, that then forms a hydroxyl group, we form lactate, and then NAD plus is being released. Okay, so now let's talk about alcohol fermentation. So I'm not quite sure if you've ever heard about this, but fermentation is just a historic term that refers to enzymatic reactions that, let's say, have been used also on a larger scale by humans for a long time, like alcohol fermentation. Okay, but it's pretty much just another term for an enzymatic reaction. So, in contrast to the lactate dehydrogenase reaction, the production of lactate from pyruvate, which is a one-step reaction, here we are dealing with a two-step process that leads from pyruvate to ethanol. And the first step actually is a decarboxylation. So we reduce the carbon number in our substrate pyruvate from 3 to 2 here in our intermediate, which is acetaldehyde. And this decarboxylation reaction just removes one carbon, this carboxyl group, in the form of carbon dioxide, and the enzyme appropriately is called pyruvate decarboxylase. Okay, so you should know the names of these enzymes and familiarize yourself with these enzyme names. I pointed out in my in my study guide that I certainly expected to know all the names of glycolysis and such. Are you studying for my exam at least? You don't have to sit here, you know that, right? So you certainly need to know the names of the enzymes that participate in glycolysis. Pretty much all the enzyme names that we discussed, you should know. But here, in this case, pyruvate decarboxylase has some similarities to the aforementioned big protein complex, pyruvate dehydrogenase complex, carries out at least this kind of decarboxylation reaction. So the other enzyme, pyruvate dehydrogenase, much more complex, also performs other reactions, but the first reaction it catalyzes is pretty much what pyruvate decarboxylase does here. It removes a carboxyl group to convert the 3-carbon compound pyruvate here to a 2-carbon intermediate, or in this case, 2-carbon product for this pyruvate decarboxylase reaction. And the product here of this reaction is acetaldehyde, and the cofactor that is utilized both by this pyruvate decarboxylase and, as we'll see a little later, also pyruvate dehydrogenase, is thiamine pyrophosphate, here abbreviated TPP. And thiamine pyrophosphate is actually a very important cofactor that is used by enzymes that cleave bonds adjacent to carbonyl groups. And often enough, TPP is said to carry active aldehyde groups, and we'll see what that means when we look at the mechanism. So thiamine pyrophosphate is just the pyrophosphorylated vitamin B1, which is thiamine, which would be this part of the molecule, and then we just attach 2 phosphates. This is a pyrophosphate. So we have a pyrimidine-based ring system here, which is linked to the important part of the cofactor, which is a thiazolium ring. So thiazolium, I'm not quite sure how sound your knowledge of heterocyclic nomenclature is, but thiazolium means that we have, in addition to carbons, we have a nitrogen and a sulfur, or the other way around, a sulfur and a nitrogen, okay, thiazolium. So this is a 5-membered ring here, where one carbon is flanked by a nitrogen and a sulfur here, and in the protonated form it has a hydrogen here linked to this carbon. As I indicated, this is an acidic proton, which means that it can be relatively easily be donated, and this is of crucial importance for the activity of this cofactor and the enzymatic activity, because it's the carbanionic form here of this carbon which is the active form. And if we deprotonate here this carbon, we get a negative charge here, and we have it right next to a positive charge, so this is a classical elide. Okay. So what does this cofactor then do? So in the deprotonated form, it's a good nucleophile and can easily attack here the partially positive carbonyl carbon in our substrate, pyruvate, and we form an intermediate where our substrate is covalently linked to our cofactor here. So this resembles, or looks like, or is actually, if we look at it like this, a derivative, a TPP derivative of lactate, we could say, okay? Now the oxidation state is different, but nevertheless, it looks like a derivative of lactate. From this here, this carboxyl group is cleaved off as carbon dioxide, and these electrons move over here, and we form a carb-anion here, which is resonance stabilized, never mind. But if we protonate this carbon, we get a hydroxyethyl, so ethane hydroxylated, okay? Pretty much an ethanol derivative linked to thiamine pyrophosphate. And then we cleave this bond and we release acetaldehyde. Okay? So what happens next, let me go back to this overview, is that the acetaldehyde is actually reduced to alcohol. So this is a classical reduction again. Our aldehyde functionality is converted into an alcohol. It's a coupled redox reaction. So the cofactor that participates in this reaction catalyzed by alcohol dehydrogenase is NADH. And NADH is converted back to NAD plus and could be used by glyceraldehyde 3-phosphate dehydrogenase in glycolysis. So humans actually do have alcohol dehydrogenase, so we can perform this reaction, but what is also important is the reverse reaction where ethanol can be converted back into acetaldehyde. Acetaldehyde in this reaction is also blamed by many people for giving you headaches if you drink too much. This is not the only factor, but it's one of the culprits, presumably. That does not mean that we can produce, we ourselves, okay, our tissues, our body, could produce ethanol from pyruvate, or let's say produce ethanol from eating bread or chocolate. We ourselves, our tissues, are not equipped to do that because we do not have this enzyme pyruvate decarboxylase. Okay. Other organisms do have that, okay, like yeast or certain bacteria. And interestingly enough, in your body, in your guts, you also have such bacteria that can actually produce ethanol. And supposedly, we always have a certain base level of alcohol in our body that is based on the fact that we have in our intestines certain microbacteria that can actually perform this alcohol production. Okay, so side note also is that this reaction is also key for bread baking. Okay, oops, sorry. How did that happen? So the carbon dioxide that is produced here in our first reaction, the pyruvate decarboxylase reaction, is key to making bread rise. Okay. And I just last semester, when I taught this stuff in my 470 class, I looked up a couple papers and I found that when you eat bread, that depends on the type of bread, you also can produce alcohol. So even if you don't drink any alcohol, but you ate certain bread types or so, this could actually lead potentially to not an elevated, but let's say a measurable amount of alcohol in a breathalyzer test or whatever these things are called. Okay, so won't get you over the limit, most certainly, but at least it's detectable, and this seems to depend also on the type of bread actually that you eat. Some are contain more alcohol than others. Okay, so let's take a look at the alcohol dehydrogenase reaction real quick. So again, we have a transfer of a hydride anion from the upper phase of the nicotinamide ring in NADH to our substrate, which is now an acetaldehyde. So the formation here of the hydroxyl group here is facilitated by zinc ions. Zinc ion that we find in the active side of the enzyme. So in yeast, it's actually a tetrameric enzyme, and in mammals, it's a dimeric. Regardless of where we find this, we have zincs complexed with certain amino acid residues, and the zinc actually polarizes this bond and makes this reduction, this nucleophilic attack by the hydride easier. So we transfer of H-, these electrons move over here, form an O-, which is protonated then, and we form ethanol. Okay? So, since you see here the hydrogens in different colors, I can already at this point talk a little bit about prochirality. So this is a stereoselective reaction, despite the fact that ethanol is not a chiral molecule. But if you were to use deuterated, NADH here, you could actually distinguish these two positions. You would have a deuterium here and a hydrogen here. So this is another stereoselective reaction. And when we are dealing with substrates or products, I should say, that are not chiral themselves, but could be made chiral easily by just replacing one of the substituents, and this is a lame definition, but that's a definition that is valid, we talk about prochiral molecules, okay? And this concept of prochirality is really limited to enzyme activity, because enzymes can distinguish different sites of molecules that are not chiral themselves yet, okay, and could make chiral molecules out of them. And we'll see another example in a little while when we talk about one of the enzymes in the citric acid cycle. But okay, here you could distinguish these two.
I had a question about the enzyme. So does the proton do nucleophilic attack on the carbonyl?
Who can answer this question?
I was confused because I thought that the carbonyl would be positive, so it's like, it seemed like positive attacking positive.
It's a hydride, so it's actually a proton with 2 electrons.
Oh, okay.
Right? It's not a proton that is being transferred here, it's an H minus. So a proton that comes, you could think of it as a proton that comes with 2 electrons, or hydrogen atom that has an extra electron. Okay, so this is a good nucleophile actually, excellent nucleophile. We're not dealing with a positively charged hydrogen cation here. Okay? Okay, so what we're going to do now, as promised before, is we'll talk about the fate of pyruvate under aerobic conditions, and everything that I'm going to discuss from now on will be relevant for the next exam. So up to this point, all the material I discussed so far, all the material I discussed will be relevant for next Tuesday's exam, and everything that I'm discussing from now on will be relevant for the second exam. Okay, so as I just said a moment ago, is that under aerobic conditions, the pyruvate is first converted into acetyl-CoA, so this is coupled to a decarboxylation reaction, so we also reduce the carbon number here from 3 to 2, and this is happening in the mitochondrial matrix. So, as I said before, glycolysis occurs in the cytosol, okay? But then under aerobic conditions, both this decarboxylation reaction catalyzed by the pyruvate dehydrogenase complex and the citric acid cycle, and then also cellular respiration happens in mitochondria. So our substrate here for the subsequent reactions, pyruvate has to be transported from the cytosol to the mitochondrial matrix. Now you may think that this is not a big deal, but transport across biological membranes is always a problem, at least when we're dealing with polar molecules. Okay, here we're dealing with a polar molecule, of course. So you may remember from previous classes that mitochondria actually have 2 membranes surrounding the interior space called matrix. The outer membrane isn't really an obstacle for too many molecules, at least low molecular weight molecules, because the outer membrane contains huge pores called porins. So these are pretty much holes. It's a little bit more complicated, but you can think of it for them as holes that allow free diffusion of molecules up to a molecular weight of about 10 kilodaltons. Okay? 10 kilodaltons is actually quite big, so we're talking size of a small protein. Okay. Here we are dealing with low molecular weight compounds that have molecular weights in the few hundreds. It is that much, okay? So they can freely diffuse across the outer mitochondrial membrane, but this only brings them to the intermembrane space between the outer and the inner mitochondrial membrane. And the inner mitochondrial membrane doesn't have these type of pores. And any polar molecule, regardless of size, requires a specific transporter to be transported across the inner mitochondrial membrane. Okay? So here for pyruvate is actually under physiological conditions negatively charged, certainly needs a transport system to be transported into the matrix from the intermembrane space. And there is a transport system that transports pyruvate in symport, meaning together with protons from the intermembrane space across the intermitochondrial membrane into the matrix. And this transporter has also different names, but one that indicates what its function is quite clearly is pyruvate translocase. Okay, so it translocates pyruvate from the intermembrane space into the matrix. And when I refer to transport across the inner mitochondrial membrane, I may use the term cytosol or intermembrane space synonymously, okay, because this outer membrane isn't a barrier. So when I say it's transported from the cytosol across the inner mitochondrial membrane, that's an oversimplification, okay, but just think it can freely diffuse through the outer membrane anyway. So once pyruvate then reaches the mitochondrial matrix, so the inner space of mitochondria, it is converted into acetyl-CoA by this big protein complex that I mentioned briefly before called pyruvate dehydrogenase, or pyruvate dehydrogenase complex. Okay, so strictly speaking, this reaction, the conversion of pyruvate into acetyl-CoA, is not part of the citric acid cycle. Okay. The citric acid cycle is, as I said before, a cyclic metabolic pathway, which is actually composed of 8 enzyme catalyzed reactions, and this conversion of pyruvate into acetyl-CoA is not part of it. However, this pyruvate dehydrogenase complex reaction is always discussed together with the citric acid cycle because it's a prerequisite if the source of acetyl-CoA are carbohydrates. And we just talked about glycolysis, we talked about the fates of pyruvate. Here we're talking about the fate of pyruvate under aerobic conditions, so we need to discuss this reaction first before we can talk about the citric acid cycle. Now, we will talk about all these steps that make the citric acid cycle in detail. We'll take a look at some enzyme mechanisms again. We'll take a look at the energetics of these reactions and the regulation of the regulatory steps and such. But nevertheless, I have this slide in here because I think it's a good overview slide here. Gives you all the structures of the intermediates, gives you the names of the enzymes and such, all the stuff that you certainly need to know. But first, we have to talk about this pyruvate dehydrogenase complex reaction, so conversion of pyruvate, 3-carbon compound, to acetyl-CoA, where pretty much our carbons that stem from pyruvate are linked to coenzyme A. So acetyl-CoA you can think of as an activated acetic acid. So we could think that pyruvate is actually converted to acetic acid, but then the acetic acid is linked via thioester bond to coenzyme A. Now, at first glance, this reaction seems to be relatively straightforward. Okay, we reduce the carbon number. It's a decarboxylation reaction, so obviously we produce carbon dioxide again. And we produce a 2-carbon compound that's just linked to a cofactor. Okay. However, this is anything but simple. It's not that straightforward. It's a rather complex reaction which requires at least 3 enzymatic activities, namely the pyruvate dehydrogenase activity. So this is part of the complex. Same name, pyruvate dehydrogenase complex. A dihydrolipoyl transacetylase activity. Okay. Dihydrolipoyl dehydrogenase activity. Now, conveniently enough, we can refer to these 3 activities as E1, E2, and E3, and that's what I usually will do. And these enzymes actually require cofactors. So this reaction, which looks so easy, requires participation of 5 cofactors and comprises a couple of individual biochemical reactions. Now, the enzyme complex is pretty big and ranges in size depending on the tissue of the organism that it's isolated from, between a couple to more millions of Daltons. It's so big, actually, that it can be visualized using an electron microscope. So in order to be visible with a standard electron microscope, a particle has to be considerable in size. And here we see that, okay, we have tens of nanometers, 50 nanometers or so at least, diameter for these complexes. And in mammals, for example, a complex is made of about 30 copies of E1, 60 copies of E2, and at least 6 copies of E3. So this is a big complex, and then other regulatory proteins can actually also attach to this complex. So it's pretty big. Okay. Now this is an overview again for you. So if you want to study this, you have everything on one slide that's happening here. I may come back to this also, but we are going to talk about all these individual reactions step by step. Also, this pretty much serves the purpose of helping you to understand what the individual enzyme activities actually do and where we find the different cofactors that are linked to these enzyme activities. So we see that we have 5 cofactors. We have a thiamine pyrophosphate, same cofactor that we just discussed for pyruvate decarboxylase, okay, remember that? Which participates in the overall conversion of pyruvate to ethanol, first step. And it's bound to E1, it decarboxylates pyruvate, okay? That's obvious. Then we have something that you can call lipoic acid or lipoic acid. I would say lipoic, think of lipid, okay, but you can also say lipoic acid, which is covalently linked to a lysine residue, okay, on E2. We'll see what it does. Then we have coenzyme A. Okay, coenzyme A obviously accepts here our acetic acid residue to form acetyl-CoA. And then we have flavin adenine dinucleotide and nicotine amide adenine dinucleotide to make this list complete. And they're bound to E3. So let's talk about this reaction, this overall reaction step by step. So as I promised, the first step is very similar, pretty much almost identical to what we just discussed for pyruvate decarboxylase. So the anionic form of thiamine pyrophosphate attacks our carbonyl carbon here. We form, after decarboxylation, a hydroxyethyl intermediate, okay? Now, we saw that in the pyruvate decarboxylase reaction, this is actually released as an acetaldehyde, okay? Here it's different because this hydroxyethyl thiamine pyrophosphate actually is given to our second cofactor, lipoamide, lipoamide. Okay. Before we come back to this, let's first take a look at this cofactor. So lipoic acid would be this part in black. If we had a carboxyl group here, you can think of lipoic acid as an octanoic acid, so a carboxylic acid with 8 carbons. That at the 6th and the 8th carbon has a sulfhydryl group. Okay, and these 2 sulfhydryl groups actually can be oxidized to form a disulfide bridge. That's lipoic acid. Okay. Supposedly, it's not, best of my knowledge, it's not a vitamin, so we can also produce lipoic acid. In the E2, so our dihydrolipoyl transacetylase activity, this carboxylic acid actually has been covalently linked to the epsilon amine of a lysine residue to form this amide bond. And therefore we can refer to this as a lipoamide or lipoamide. Okay. Remember, conversion between disulfide into sulfhydryl groups is a reduction. Okay, we only have coupled redox reactions. So if we convert the oxidized form to reduced form, something else needs to be oxidized. Okay? And this actually happens when the hydroxyethyl residue linked to thiamine pyrophosphate is transferred here to one of these sulfurs, and we form the acetylated form of lipoic acid. Okay? So if we go back to this reaction here, as this transfer occurs of this 2-carbon unit to this sulfur, we have a redox reaction occur, and we form what kind of covalent bond here when the 2-carbon unit is linked to lipoamide? What could it be? Just think about what I just said. We have an oxidation. What was this? In the pyruvate decarboxylase reaction, we would cleave off what? We would release what? Here from TPP? What did I just say? Oh, come on. So stupid. Are you so overwhelmed? Come on. What is released in the pyruvate decarboxylase reaction that we discussed 10 minutes ago?
Carbon dioxide or CO2?
What?
Carbon dioxide or
Yeah, carbon dioxide and go back to your notes.
NAD plus.
That doesn't happen in the pyruvate decarboxylase reaction. It happens in the alcohol dehydrogenase reaction. So what happens in the pyruvate decarboxylase reaction? Go back to your slide. Tell me, please. I want you to make this conclusion. Gotta be kidding me.
The CO.
What is it? I mean, look at the first reaction. Come on. Can't be serious.
Acetaldehyde.
Acetaldehyde. Okay? So this carbon here has the oxidation state of an aldehyde carbon, right? Right? Okay. Now what happens if we oxidize an aldehyde? What did I tell you about 2 dozen times? It's oxidized to? Please, I'm begging you. To carboxylic acid, right? Okay. So let's go back to this reaction. I said that here the transfer of the hydroxyethyl group to our lipoamide is coupled to a redox reaction. The disulfide here in our cofactor is reduced, so something else is being oxidized, right? And it's the carbon that before it's being transferred is still in the oxidation state of an aldehyde. So what will be the oxidation state afterwards, most likely? One of a carboxylic acid. Now if we have a carboxylic acid linked here to a sulfur, what is the functional group that we have here? Guys, you have an exam on Tuesday. You realize that, right? A thioester, of course it's a thioester. Okay? I mean, what else would it be? So, we now have a 2-carbon unit that actually has a hidden carboxylic acid in it, right? Now the acetylated lipoamide actually then is used to transfer this acetyl group to coenzyme A, okay, to produce acetyl-CoA, which leaves us here in our transacetylase with a reduced form of dihydrolipoamide. Okay. In order to repeat this enzymatic reaction, we have to regenerate the oxidized form here. We have to introduce a double bond here again. And pretty much the other reactions that we see from now on just serve the purpose of regenerating the oxidized form here of our sulfates. Because at this point, we already have produced acetyl-CoA, right? Okay, so, not sure if you've ever seen the chemical structure of coenzyme A before. Not sure, did Dr. Baker talk about this? No? Yes? No? Okay, doesn't matter. So, coenzyme A, here we see the acetylated form. Okay, in the deacetylated form, we would just have an SH here, a thiol group here, is an important cofactor for the transfer of carboxylic acids. Okay. So, the most commonly known form is an acetylated coenzyme A, where coenzyme A is used to transfer acetic acid. And therefore, we can say that this is activated acetic acid. That's a term that you see in the literature occasionally. However, when we talk about lipid metabolism, we will see that coenzyme A and, let's say, derivatives of coenzyme A actually also are important for transferring other carboxylic acids, namely, in particular, fatty acids. Okay, so the nomenclature is such that usually you wouldn't refer to a carboxylic acid as a fatty acid if you have fewer than 4 carbons or something like this, but in principle you can think of it, okay, transfers carboxylic acids, fatty acids. Now we see here that actually a part of this cofactor is a phosphorylated adenosine, so we have an adenine here linked to a phosphorylated ribose at the 3' position. Then we have a pyrophosphate bridge to this structure, which is pantothenic acid. This is a vitamin, so we have to take it up with our diet. It's vitamin B5. And then via an amide bond, this pantothenic acid is linked to the part that is really relevant for this transfer activity, transfer of carboxylic acid. You can give it different names. One is given here, it's a beta-mercaptoethylamine. Okay, sounds more complicated than it actually is. It's based on an aminated ethane. Okay, so it's 2-carbon unit with an amine. The amine here has formed an amide bond with the pantothenic acid, and it's called beta-mercaptoethylamine because in the beta position to this amine alpha beta, we have a sulfur, and sulfur and sulfhydryl groups here can also be called mercaptanes. Okay, so it's a mercaptoethylamine, and it's here that coenzyme A can actually accept or donate from carboxylic acids that can be linked to this position again via thioester bonds. Okay, so this happens here in this reaction catalyzed by dihydrolipoyl transacetylase. The acetyl group linked to lipoamide first is transferred to coenzyme A, we produce acetyl-CoA, and then this reduced form of our cofactor lipoamide, which we can call as dihydro because we have 2 hydrogens now at the sulfurs, lipoamide. So, as I said before, the next reactions simply serve the purpose to reoxidize these 2 sulfurs and regenerate the disulfide bridge. Now, E3, our third dehydrogenase activity, the third enzymatic activity actually, has a disulfide bond, and it also has a flavin adenine dinucleotide, so it's a so-called flavoprotein. And both of these can carry out redox reactions or participate in redox reactions. So what happens first is that our 2 sulfhydryl groups in our lipoamide, dihydrolipoamide, are oxidized with the help of this disulfide bridge in E3. So we have formation of a disulfide bond again in lipoamide, and we have now 2 sulfhydryl groups here in our third enzymatic activity. Now we have to regenerate that also. We have to produce our oxidized form again so that we can continue with this enzymatic activity. Here this flavin adenine dinucleotide, which is also bound to E3, plays a role because it will actually oxidize these 2 sulfhydryls, okay, and be reduced itself. This won't do us much good in the long run, because if we want to continue with this enzymatic reaction, we have to oxidize this flavin adenine dinucleotide back, okay? And we can do this by utilizing NAD+, which is reduced to NADH when this FADH2, the reduced form of flavin adenine dinucleotide, is being oxidized, okay? And overall then, in our pyruvate dehydrogenase complex reaction, pyruvate is converted into acetyl-CoA, and NAD+ is converted into NADH. So we have actually production of an energy-rich compound here also in this decarboxylation reaction. Oxidative decarboxylation is also called because it's coupled to a redox reaction. Okay, so here is the structure of flavin adenine dinucleotide. You may or may not have seen this in other classes. So it's an analogy to nicotine amide adenine dinucleotides, so the dinucleotides. So we should be looking for 2 nitrogenous bases, 2 sugars, and 2 phosphates. Similarly to NAD+ or NADH, one of these nucleotides is simply an AMP, an adenosine monophosphate. So we have an adenine, a ribose, and a phosphate linked to another phosphate, linked to another sugar, which is actually a reduced form of ribose called ribitol. And this sugar is linked to another nitrogenous base, which is neither a pyrimidine base nor a purine base. But it's called isoalloxacin ring. Okay. So here on the left side, we see the oxidized form of this cofactor. We also are looking at the dinucleotide, so there is a sister compound of the dinucleotide, which is simply the mononucleotide. Instead of FAD, flavin adenine dinucleotide, we can call it flavin mononucleotide, which lacks this AMP portion. Certain oxidoreductases that are also called dehydrogenases use FMN, others use FAD. With regard to the redox reaction, they are the same, okay? And all the redox activity actually resides here in our isoalloxacin ring. Typically in textbooks, the reduction and oxidation, which is reversible here for our isoalloxacin ring, is shown as shown on this slide, which shows that this can happen in a stepwise manner. Okay, so where first one proton and 1 electron are taken up, one position here of our isoalloxacin ring to partially reduce this ring system. So we form a semiquinone, the radical here. And then in another uptake of an electron and a proton, we convert the semiquinone to a fully reduced FAD, which we can refer to in abbreviated form as FADH2, or if this happens to the mononucleotide FMNH2. So this is a reversible process. So FADH2 or FMNH2 can also donate electrons, 1 electron at a time. Okay? And this will be more relevant actually when we talk about electron transfer, as it also occurs in mitochondria, where single electrons can be transferred by a bunch of appropriate cofactors and ions and such. Okay. What we are going to see actually when we talk about the electron transfer chain is that there is a flavin adenine dinucleotide at the very beginning of this electron transfer chain that accepts electrons from NADH. And I told you that NADH actually is oxidized to NAD+ by donating a hydride. Okay? And a hydride, again, is a hydrogen that contains 2 electrons. So this is the transfer of 2 electrons at a time. Okay? And NADH is actually the only cofactor that in the electron transfer chain, as we'll see and discuss in more detail soon, transfers 2 electrons at a time. So the acceptor in this reaction is also a flavin adenine, mononucleotide, I believe, which then has to be capable of accepting 2 electrons at a time. So this representation will not cut it, will not show it, will not explain what's happening actually, and therefore I have added actually another representation that shows that we can also have 2 electron transfer from NADH to this isoalloxacine ring, but both transferring electrons at the same time. And this is shown here, okay? Because that's what you usually see in textbooks, but that doesn't tell you the whole story. I skipped this slide because we talked about the structure of NAD plus and NADH and also the phosphorylated version of these last time when we talked about glycolysis. Okay, so this is the reaction that we now spent 15 or 20 minutes talking about. It's not that easy. It comprises at least 3 enzymatic activities and at least 5 cofactors. It's relatively complex, but if you think about it and see the analogy, for example, between what happens in the pyruvate decarboxylase reaction and in the decarboxylation here that leads to acetyl-CoA production, it's not that complicated anymore. Okay, so this acetyl-CoA or acetyl-CoA that is produced from the degradation of other biomolecules, lipids and proteins and fatty acids and amino acids respectively, can be channeled into, under aerobic conditions, into the citric acid cycle. The citric acid cycle has been given a number of names: tricarboxylic acid cycle, for example, because there are a couple of tricarboxylic acids actually that are intermediates of the cycle, or Kreb cycle after Hans Krebs, who was a pioneer in biochemistry and elucidation of this pathway. I will usually call it citric acid cycle. Okay. So this again is another representation of the citric acid cycle. Again, when you come from degradation of carbohydrates, a prerequisite would be the pyruvate dehydrogenase complex reaction, which strictly speaking is not part of this complex and this pathway, excuse me. And the first step here is the transfer of the 2 carbon units, the acetic acid unit that we find in acetyl-CoA, to a 4 carbon molecule called oxaloacetate. I just told you that the citric acid cycle is also called a tricarboxylic acid cycle because there are a couple of tricarboxylic acids. For that, you may want to remember what a dicarboxylic is, what a tricarboxylic is. So a dicarboxylic acid would have 2 carboxyl groups. Tricarboxylic acid, analogously, would have 3 carboxylic acids, right? If you look at the different intermediates here in this cyclic pathway, you will actually see that there are many more dicarboxylic acids than there are tricarboxylic acids. And I always, when I discuss the citric acid cycle, I always encourage my students to familiarize themselves with the names of the simplest dicarboxylic acids, because it helps you a great deal with remembering the names of many of the intermediates of the citric acid cycle. It also helps you relating names to the structures of these molecules that are intermediates of the cycle. So the simplest, my students who took 470 may remember that, the simplest dicarboxylic acid would just be 2 carboxylic acids linked to each other, right? You know what this molecule is called? Anyone remember? It's called oxalic acid. Okay, you may want to write this down because that's actually quite helpful, I guess. And we see that, okay, we have one of the starting materials of the first step actually being called oxaloacetate. Okay, think of this as an acetate derivative of oxalic acid. The next dicarboxylic acid would have a CH2 bridge between 2 carboxyl groups. Okay, do you know what that is called? Any idea? It's called malonic acid. Now, malonic acid is not relevant here for the citric acid cycle, but we'll see a derivative of malonic acid actually when we talk about biosynthesis of lipid acids, fatty acids. Excuse me. So the next dicarboxylic acid would have 4 carbons. 2 carboxylic acids flanking CH2, CH2, right? Any idea what this is called? Any guesses? Succinic acid. Okay. Succinic acid, don't see anyone write this down, okay. We see actually not only succinic acid or the ionized form of succinic acid called succinate here being true intermediate of the pathway, but we'll also see an activated form of succinic acid or succinate, where succinate is linked to coenzyme A here being an intermediate, succinyl-CoA. So it's simply a derivative of the 4-carbon dicarboxylic acid. If we move on to 5 carbons, we're dealing with glutaric acid. We actually have a derivative of glutaric acid here in the citric acid cycle, a glutaric acid that in alpha position to one of these carboxyl groups has a ketone, and therefore we can refer to this compound as alpha ketoglutarate. Glutaric acid in the ionized form is glutarate. Okay, so if you know these simple dicarboxylic acids, it can relate the names of these dicarboxylic acids to their structures, which should be simple enough. You can easily deduce the structure of a variety of these compounds that you have as intermediates here in this cyclic pathway. Okay, a couple of things here that I want to mention is that a 2-carbon unit in the form of this acetyl group linked to coenzyme A enters the cyclic pathway. Okay. We have 2 oxidative decarboxylation reactions in the cycle where carbon dioxide is released. So from a stoichiometric point of view, a 2-carbon compound enters this cyclic pathway, and 2 1-carbon units actually leave the pathway in the form of carbon dioxide. If we look at these red boxes here, we will see that it's not the very same 2 carbons that enter the cycle that also leave the cycle, at least on the first round here, in the form of carbon dioxide. But from a stoichiometric point of view, we have a 2-carbon compound enter the cycle and 2 1-carbon compounds being released. Okay. Another important thing here is that in this cyclic pathway, a number of energy-rich compounds is being produced. It's either ATP or GTP. We'll talk about what might be produced, but basically an ATP equivalent is produced in one of these reactions here. And then 3 molecules of NADH and 1 molecule of FADH2 are produced in other reactions. So remember that NADH and FADH2 are energy-rich compounds. So this happens under aerobic conditions where we can also have cellular respiration, where the energy stored in NADH and FADH2 respectively can be utilized for the production of more ATP. So just going through this cycle here once actually produces quite a bit of energy. So this is a very important cyclic pathway with regard to energy production. Okay. For that reason, citric acid cycle is typically discussed in the context of catabolism, energy-producing metabolic pathways. We will see that, and I mentioned this briefly before, I think one of the first classes, that the citric acid cycle is also important for anabolism, for biosynthetic reactions, because quite a number of these intermediates here can actually be utilized as starting materials for the synthesis of other biomolecules. So it's amphibolic in nature, the citric acid cycle is. Okay. But from an energy production point of view, a lot of energy carriers are produced here. We'll talk about the citric acid cycle in more detail next week. How will we do?